with a crowd by Brother Salim from our office.
أعوذ بالله من الشيطان الرجيم بسم الله الرحمن الرحيم إذا جاء نصر الله والفتح ورأيت الناس يدخلون في دين الله أفواجا فسبح بحمد ربك واستغفر إنه كان توابا صدق الله العظيم.
Jazakallah khair, Brother Salim. Alhamdulillah, today we have joined for the workshop on preparation of science fair projects, particularly the science fair projects in biological sciences. Respected Dr. Qazi Azhar Saab, who is the advisor, is the mentor for the science fair we are conducting over 15 years. Jazakallah sir for having joined the program.
Assalamu alaikum.
Our Joint Secretary Dr. Nihar Akhtar Madam, our resource person Dr. Saquib, Assistant Professor of Zoology, New College, and all our beloved teachers. Assalamu alaikum wa rahmatullahi wa barakatuh. Good evening to one and all. In the series of workshop to train the teachers how to guide the students to do the science fair projects following the scientific research methodology. It is a third week we are conducting. Today, the topic is the preparation of science fair projects in biological sciences. It is going to be, the session is going to be handled by Dr. Saquib Naveed. Actually, we are very fortunate to have Dr. Saquib Naveed with us. He is a very active researcher, young scientist from New College, where I worked and I have retired. Alhamdulillah, he has published more than 30 research papers in national and international journals. It's not easy to publish a research paper at national and international level. About that, Dr. Saquib Naveed is a reviewer. He reviews the article for the publication. He has to judge whether the part an article, a research project article, is worth publishing in the international journal. He is a reviewer in the three renowned journals: Journal of King Saud University Science Elsewhere, Regional Studies in Regional Studies in Marine Science, that is also Elsewhere publication, and the International Journal of Zoology and Applied Biosciences. Such a renowned scholar we have with us today, and he is a active researcher. He is guiding the 4 research scholars for the award of PhD. He has guided more than 16 projects for the award of MSc, and more than 15 years of experience as an assistant professor. And we are very fortunate to have such a great scholar with us. And not only that, he is actively taking part in our science fair programs. He has been as a judge for our research plans and research research final papers for the selection as well as for the judging the science projects in the in our national science for the awards and prizes. We are happy that Dr. Shakib Naveed has accepted to conduct this session. Now over to Dr. Shakib Naveed.
Assalamu alaikum, sir. Thank you for the
Walaikum
kind
salam.
words, sir. I thank the
Yes, sir.
body for giving me this
Sir,
opportunity.
you just increase your audio, sir. Not
Is I am audible to you now, sir?
audible, just increase the voice.
Okay, sir. Is it
Audio.
okay, sir? No?
No, okay.
Okay, sir. Thank you, sir. Thank you for your kind words, sir. I mean, you have used many kind words for me. Thank you, sir.
Okay.
I first of all, I'd like to thank the OMIET organization for giving me the opportunity to be here. I thank the I really Assalamu alaikum, sir, the Haji Azhar sir. I take the
Walaikum
opportunity-
salam.
Thank you, sir. I take the opportunity here to thank all the people who have come here to hear me. The webinar for science teachers on the preparation of a science fair projects, National Science Fair 2020-23. Shall I start my PowerPoint?
Yes, sir. Yes, sir.
Yeah, I start sharing my screen, sir.
Your co-host, you can share, sir.
Yes, sir. Are you able to see my screen, sir?
Yes, sir. Yes, sir. Better you go on slideshow, sir.
Sure, sir.
Yeah, very good.
Shall I turn off my camera so that it will be a disturbance for you people? Okay. Can you see my screen, sir? No?
Yes, sir. Yes, sir.
Thank you, sir. Thank
Clear.
you, sir. Thank you, sir. Let us first know why we should have a science project in school. I hope I am addressing teachers here. And is there any
Yes, yes.
students here, sir?
No, no, only
Only
teachers.
are there. Okay, sir. Thank you, sir. So why should we have science project in school? As said by Haji Azhar sir, I saw his book on science project, and he has used very good words to explain why we should have it. See, I'll start with one simple thing. Usually in the childhood, the children ask so many questions, so many curiosities there. So to inculcate that talent in them, we need to have this project. And also sometimes when we give some kind of toys or some kind of machines, or they try to break it open and see how it works mechanism. It is inherent in the children's actually. And this tendency we need to foster them and kindle them to be a scientist in their inshallah. So the science projects bring out the innovative talents which is there in them, and gives them an opportunity to have a hands-on experience in scientific research studies. This is more important, which gives them confidence also. And while handling these experiments, they start enjoying it. So the scientific research is joyful experience for them. And if they start enjoying what they do, I hope that may lead to some new discoveries, innovative technologies, and inventions. and many of them may become even entrepreneurs also. Shall we move on to say next slide, sir? So why, before starting a project, we need to have planning before we're doing any kind of things. It all begins with the field of or a sub topic what we decide. So it must be realistic in terms of time available and a level. Maybe hardly we may have some 2 or 3 months to do this project, and we cannot have that project which may run for months or years sometimes. Similarly, we have to also see the level, at what level the student is. According to that level, we have to kindle them. Otherwise, it will be too less for them, or sometimes it will be too more for them. So, and always we should keep the aim of the project simple as it can, because we need to investigate one single factor at a time. Putting many factors, it may create complications in them to study the project in a better way. So before making that project, we need to have a hypothesis. Hypothesis is hypo means before, and thesis means writing. Before you, what you are going to write is that.
Sir, the volume is still low, sir.
Still low, madam? Sorry.
Yeah, you can increase it in your microphone.
Sure. ma'am. Is it okay now?
Yes, yes.
Okay, thanks. So we should ensure the experimental test measurements carried out are accurate and reliable, because these experiments need to be repeatedly, and it should be valuable. It should be validated. So before we start doing this project, we have to understand what are the controls we are going to have, what are the variables or independent variables or dependent variables we are going to use for this test, and how many variables we are going to study. We also need to correctly identify the key variables, the cause and effects, independent and dependent variables. What causes what? For example, in the case of, if you see the obesity in Indian population, what could be the reason for this obesity? Is it because of a lifestyle? Is it because of feeding habit? Is it because of the genetics? All these things we need to study. Okay. So how many repetitions or what we say, these trials or repetition observations of samples are required? Replicates. The simple terms replicate we use for these scientific experiments. How many replicates we are going to use? If you're going to use more replicates, it's more dependable, it's more authentic, the data will be more authentic. If you use lesser replicates, the bias may be, errors may be more. So we need to decide how many number of replicates we are going to have, trials in the experiment. So what kind of experiments, instruments, or equipments, or the technique we are going to use to obtain the relevant data? This also need to be pondered over before we start doing these projects. So identifying the suitable material and equipment to be used. If the investigation requires use of a questionnaire, before asking or starting the survey, we should design the questionnaire in a proper way. The questionnaires should have proper grammar to avoid redundancy and paraphrasing same words, and also to standardize before implementation. because you ask something and they think something else. That kind of situations we need to avoid in the questions. Is it the intended procedure safe and ethically permitted? For example, you don't tease any kind of animal, or you don't stress any kind of animal. All these things are there. Nowadays, this animal ethical society is very strict, and we need to get period permission before we start doing on vertebrates. Invertebrates we can do, but vertebrates we need to start ask permission before we do any kind of trials on vertebrates. So how data will be collected? How much data? What is the quantity of data? All these things does matters. So these are the few questions which need to be ponder over before we start the project. So there are some basic steps in research projects, basically identifying and developing your topic. Selecting a topic is one of the most challenging part of the project. Your topic should be standing out from your other students by selecting an interesting and off-beaten path topics you can use. The more creative the topic is, the better will be the result will be. Before we start the experiment, we need to have that background data of that topic. We have to have enough information about the topic. If you find a lesser information, you need to broaden the topic. If you have more information available on that particular topic, then you can focus on one single aspect of that topic. So we can see is through encyclopedias, dictionaries, catalogs of books, scholarly journals, and other online sources. Locating the material is one of the tricky tasks. Nowadays, we have got this alpha catalogs where you can see what is the work which already been done on the topic. Is the work what we are doing is same, the repetition? Are you doing the duplication of the same work? That kind of things can be avoided if we do some kind of homework, whether this experiment is really has been done, or is there a smaller change in the variables? All these things you can tweak the experiment with the help of these alpha catalogs and other things, books, search engines like Google, Yahoo, Bing, DocPel, DuckDuckGo, and etc. You can also look for information on this topic through electronic periodicals, magazines, newspaper, articles, scholarly journals, other things. Nowadays, this is the era of knowledge bombarded on us. Most of the, wherever we go, we have on the fingertips, we have informations. But how reliable the information what we are getting is, that is matters. We need to evaluate the source of the informations. The informations what we are gathering, is it really credible or authentic or reliable, or its accuracy? How accurate is the data is? If it may be accurate for some 30 years back, but now it may be absolute no, because of the advancement in the technologies. Similarly, reasonableness, fairness of the data, or consistency of the data, what we are getting is also important. And does it support or corroborate or agree with the previous agreements or results, or it disagree with the previous results? These things we need to check before we start collecting the data. So the C stands for credibility. A stand for accuracy, R stand for reasonableness, and S stand for support. The CARS checklist for information quality need to be done before we start gathering this information on the particular topic or subject. We also need to make notes, need to maintain a journal of our work, observations, what we observed during the project, that have to be recorded in the journal. So these informations help us to arrive at the results and infer the results. So we need to note down the information and prepare for the project based on the journal. Then followed by documents all the sources, creating bibliography or references. If you don't give references properly, it may lead to plagiarism. That is, you don't give credit to what you are using. And last part of it is writing your report, which is organizing the informations, preparing a draft, a rough draft, revising the paper. That I'll go into details in the later slides. And citing your sources properly. If you don't cite your sources properly, you are indulging yourself in the plagiarism.
Sir, still, can you increase your volume? Audio? Check possibility.
Pardon me, sir?
At least you can speak a little louder, sir.
Yeah.
Okay, ma'am.
Voice
The
is
voice
still
is
low.
very feeble. Yeah.
Yeah.
Okay, ma'am. So writing the paper, how it should be maintained. It is basically it is called IMRAD or IMRAD. I stand for introduction, M stand for materials and method, R stand for results, and D stand for discussion. In the title, before you give the title, it should be catchy enough to grab the attention. You should not start with the studies on so and so or something like that. Studies on so and so or empirical studies on something like this. You should not. You should avoid giving such kind of titles. The length of the title should be optimum. It should not be too short or too long. And a proper placement of the word should be there. That is syntax. You say something and they understand something. It should not be there. So syntax should be taken care when we give title. It is usually capital letters are used for the title, and to avoid complicated grammars and redundancy. You don't repeat the same words in the title. We should avoid. Coming to the abstracts and keywords, abstracts gives you before we start reading any kind of paper or kind of any project, we look for the title. If the title is impressive, we read the abstract. If the abstract is impressive, then we go for the full paper. We give a read to the full paper. Otherwise, there itself we will discard the paper. We don't read that paper anymore. So abstract should be brief as it can be. It should have the summary of the work done. The whole things put up in 200 words. It should also highlight the salient feature of the your hypothesis, or any kind of new methods you might have used for the methodology, or some kind of results which are very impressive results. And if application of the results is something can be applied in the physical and practical applications, that can be highlighted in the abstract. It also should be in a single paragraph. Keywords are usually given for any kind of a project work or a research work to facilitate in searching the work. It should be not more than 3 to 4 words. Coming to the introduction. Introduction is you first start the introduction with a very broad field of area. Then you slowly narrow down your field to the hypothesis or the work what you are going to introduce. So you become very specific. First, it is very broad. Then you slowly become specific to the work. That kind of talent should be shown in the writing the introduction. You also try to justify the choice of the specific methodology. You might have, there may be same work might have been done using different methodology. If you're going to use some kind of new equipment or some kind of new methodology, or you have picked the experiment with some kind of a new instrument, that can be highlighted in the introduction part. And you also introduce any kind of definitions you used or abbreviations you used in the paper, which is being repeated, will be repeated in the paper. These things we need to keep in mind when we write the introduction of the paper. Similarly, when we present the work in any kind of science fair, we need to show or stress that the work what we are carried forward with what we have done is needed work. It is not just for the sake of a science fair it has been done. It has importance or applications or has some kind of implications in the theoretical implications or practical applications. Such kind of justifications should be given when you introduce the science project or a scientific work or an experiment. Apart from this, you can also highlight any kind of gap in between the subject which you are going to fill up with these experiments or this project. Coming to materials and methods, here you give detail of the materials and methods used in the project. Why we need to furnish the details means it should be reproducible. Any scientific work should be reproducible. I do the same experiment, you do the same experiment, I get different result, you get different result. That's not going to happen. So to have replication or reproducible results, we need to give details. Of course, you may, or sometimes we may go for patenting of our work. Then we can, till the patent has not come out, we cannot, we should not give, we should not publish our work. So here in the case of materials method, you specify if there is some kind of a survey, then you specify what kind of target audience were there, the respondent, who are the respondent, what is the age of the respondent, what is the gender of the respondents, what is their socioeconomic status of the respondent, or some kind of educational qualification of the respondents or income. All these things you can include or furnish in this materials method. If you're going to design a kind of experiments, then you can say what kind of experiment is there, what kind of instrument you used, what is the make or a brand you use. You can even specify the details of the equipments or instruments. Similarly, if you try to give do kind of some kind of field studies, then you have to give the place details, what is the date you have done the sampling, where you have done the sampling, how you have done the sampling, how many samples you have done, how you collected the samples, what is the things you used. Did you use any kind of quadrants, or you use some kind of net, do you use some kind of corers, or you use just like that transects, any kind of what kind of techniques you use for collecting the samples, how you collected the samples. Did you use any kind of special instrument to collect the samples, or just like that you picked up? All the details you need to furnish in the materials method. Then it also ascertain that methodology is appropriate. It gives an opportunity to see judge the methodology, whether you have used a correct methodology for collecting that samples or response, or some kind of error has taken place. That you can see in the case of materials and methods. Coming to the results or the observations, it should be very clear. The observations should be clearly mentioned. We should avoid giving raw data here. Results, we don't need to give raw data until unless it is data is very less. If the data is very less, then you can express in a descriptive way. Otherwise, there is no need to give any kind of raw data. You analyze and then you can furnish the data in the results. You process the data, you analyze the data, then you summarize the data or distill the data and present the findings in the results section. As I said, if the data is very less, then you can express in a descriptive text. Also, you should highlight or emphasize the findings. What kind of findings you have found? Some new findings you have found. Then you can express here. And we need to avoid the redundancy. Redundancy means you give the data here in the table, same data in the figure, and then you also describe that data. So you are repeating the same result here, same data. You are repeatedly giving the same thing. This should be avoided here when you furnish the results. Mostly data should be presented in table. You can highlight the findings or values in the table, and the table should have legends there, which should be self-explanatory. The values should be of a font size of the value should be inappropriate, so that you don't over increase or over decrease the font size of the table. If you have more data which may confuse the reader, then you can graphically show the data or express the data in case of figures. Again, choosing the right figure also matters. What kind of figure you are going to use to express your data visually. This I will discuss in the next slide, what kind of figures we can use. The tables, the figures should have proper captions or legends. It should be self-explanatory. And if you give in descriptive, you should mention on which table you have given this data. the results. There may be so many tables you may have in your results. If you don't furnish properly or don't address the issue, what is the use of giving so many, furnishing so many tables? So when we present the results visually, this is what I want to show you. You have to choose a right figure to express visually the results. If you are going to compare, then how many variables you are going to compare? If the variables are few, you can use this bar diagrams or vertical bar diagrams chart. If there are many, then you can use line diagrams. And similarly, if it is one single time or over the period of time means, then you can use the chart, line chart or bar chart, vertical chart. These things you can use. Coming to relationship, if you want to show there is some kind of relationship between two variables, then you can express this in the case of a scatter plot or a scatter plot bubble size, which may show. Even you can use cluster diagrams also to show the relationship between or closeness or similarities or dissimilarities between these variables. Similarly, if it is a distribution, again you can use a scatter plot or a line histogram or a bar histograms. How it is distributed, what is it is platykurtic or leptokurtic or mesokurtic, the distribution is, what kind of distribution is. If it is composition, different components are there. If you take, for example, some sampling you have done in field sampling, then you come across different types of organisms. Then you can express them in a pie diagram. It gives so much of information in one single diagram. Which organisms are dominant in that area, which organisms are very less abundant there, are scarcely present there. And if it is dominant, to what degree it is dominant. A pie chart will give you a very good information about this distribution or composition of that place. Similarly, you can use this stacked area 100% chart or stacked area chart if it is over a period of time. For example, you have done some kind of sampling during month January, February, March, April. You can use this area, this diagram, relative and absolute difference matters. The stacked area chart or a stacked area, 100% chart you can use. Or a simple line diagram also do. So it is not only generation of data, but also how you present your data does matters. You might have done so much of work, so much of planning in generating or doing the experiments. And if you don't present your data in a proper way, it is not going to reach to the public. So we need to present it in a better way, in a better manner, so that it can be understood by anybody. So presenting data is more important. Even if it is on a paper, you may able to give it in a chart or some kind of diagrams. But if it is PowerPoint presentations, then you have to express it properly. You have to address it. So discussion, what we discussed in this is the last part of the write-up. After this, the bibliography or references comes. So we emphasize the key findings in our work or project and their interpretations. It also shows our it can agree with the previous results, or it that is, or corroborate with the previous results, or it will disagree or contrast with the previous work. We also discussed the theoretical implications and practical applications of the work, what we have carried out, and conclude clearly. And if there is a scope for the future course of work necessary, we can give a roadmap or some kind of lead to show what kind of work can be carried out in future studies. These are the things we should be discussed in the discussion. Coming to the references part, the citation format should be properly handled. There are different types of citation formats, reference format or bibliography formats. Council of Biological Editor or Biological Association CBA was there. It is now called Council of Scientific Editor, CSE. And Modern Language Association for linguistic references to handle. American Psychological Association for economics and sociology related references. Chicago or Turaban for religion, arts, and other things. We use different formats of references for different stream of studies. If we fail to properly cite the sources, that may lead to plagiarism, because it is the intellectual property of that author what we are citing here. We need to properly handle the reference part. If you don't properly handle the reference part, what will happen? It will show plagiarisms, and ultimately you may not intentionally done it, but unintentionally mistakes might have cropped in into your work, and as a result of that, that will give you some kind of negative impact on your work. So citation should be properly handled. If you are going to quote same quotes, you can use inverted commas, and you can verbatim, same verbatim you can use verbatimly under the inverted comma, and you can quote the source. If you are going to paraphrase it or going to such kind of things, you can have to give the citation properly. With regard to biological projects, based on the level of the students and his intellectual and his capability, and how much he can afford the facilities present in the laboratory, we can design the project. We cannot have a fancy project which may include expensive use of expensive or very advanced or sophisticated instruments. Or what is the use of doing a project if you outsource the analysis or test in the labs outside? So any kind of project which facilities, with the limited facilities in the schools and at home, should be kept in mind. Based on that, we have to design the project. We don't need to go for very advanced scientific projects here. It is only the necessary, the need here is to kindle the minds, the little minds, small minds here, student mind, so that he get inclined towards scientific research, start doing on his own. For that, we don't need to spend so much hefty money outside or we should not give our results analysis outside, for example in the labs, or some kind of nowadays you see many come up with the project outsourcing. Such kind of things should be avoided. So it should be realistic, simple one, simple project. It should be more creative. The project should have a creativity in it, and the project should have clarity. The experimental plan should be simple. At the same time, the controls, what we are going to use, should be properly addressed. Data collection should assure that it is reproducible. And we can go for some kind of analysis or statistics to add on the results aspect. Importance should be given to the creativity of the project or innovations of the project. How innovate it is, what is creativity, what kind of creativity is involved in this kind of project. That should be addressed properly. For example, air pollution, you can use so many things you can use. For example, I'm just trying to give you a few examples here. Air pollutions, there are different types of pollutions nowadays. Microbes are suspended in the air. You can check what kind of microbes are present if you take the sample of the air. With a simple agar plate, you can check what kind of microbes are present in a school compound, a class compound, or in the home, at kitchen, or in the bedroom. They can check the air quality using microbes, different molds. Similarly, dust, they can check what kind of dust they have, how much dust is there, all these things. What is the source, how it can be addressed. Similarly, bioremediations, you can use how this water, or mostly it is we see many of the students use detergent water, how it can be used or put to use out of bioremediation, all these things.
Sir, excuse me, sir.
Yes, sir.
Sir, still your voice is low. You keep your microphone nearby you, and or you raise your voice.
Okay, sir. Okay, sir. So you can also study the effect of various toxic substances on organisms. Effect of some kind of detergents on the fish, or some kind of things. You can use chemicals. Simple experiments. You don't need to go for a very high-tech experiments. Simple experiments you can do with the help of fish. You can see most of the schools have microscopes. You can take small photograph of their gills, how the gills are affected. If there is any lesions on the skin of the fishes, you can address, you can see, or any kind of damage to the fins, that can be seen visually under a microscope, compound microscope, or a dissection microscope. Effect of global warming, you can do some kind of experiments where you can increase temperature of that water or habitat and try to see how the animal respond to it. Similarly, phytoremediations using a plant extract, how you see effect of a plant extract in removing toxic chemicals and kind of things. Similarly, water filtration, you can use biofiltration. What kind of organisms are best suited for biofiltration or bioflocking, we say. This you can study. This doesn't need any much of equipment. One microscope will do, and little bit of understanding of morphology of different organisms, smaller organisms, microscopic organisms will do. They can take nowadays many mobile camera has got very good mobile phones has got many good very good cameras that can be kept on the microscope, and a picture can be taken out. Similarly, how the water pollution can be studied, different kinds of pollutions studied with the help of different instruments or different organisms. Most of the organisms, there are many organisms which are bioindicators. The presence or absence of the organism itself shows what kind of pollution is there. So you can make use of this kind of literature to know what kind of pollution is there. Similarly, health science, you can see you can check the BP, or there's sometimes some experiments involving simple BP apparatus nowadays comes BP monitors. Sorry, you can make use of this BP monitors and see is there any effect of BP in the activity. If it is activity, what kind of activity, or the age, or the gender. These things can be done. Similarly, vitamins, impact of vitamins on the reproduction of fishes, you can see under silagate or any kind of minerals you can use which enhance the reproductibility of the fishes. These things can be done. So apart from this effect of light on the bacteria or algae, if it is light, and what kind of light, what is the wavelength which is giving high photosynthetic activity, all these things you can see. Similarly, impact of bacteria on fungi, effect of temperature on bacteria and fungi, you can see. You can conduct small experiments. Various at home you can collect the samples, bacteria, fungi samples near the fridge, inside the fridge, near the kitchen. All these things doesn't need very costly equipments or one agar plate, a simple culture medium, and small incubator size where we can culture these microbes, and then a little bit of knowledge about microorganisms. Microbes will do. Similarly, you can study the tropism or germination of seeds. You can just hold the seeds, put in a tissue paper, leave it in a warm place, and then try to turn the tissue paper in different directions to show how this tropism is taking place. And similarly, effect of fertilizers or vitamins on plant nutrition, plant hormones, phytochemicals. What are the phytochemicals? Antimicrobial activity of plants. All these things can be done. Even this, or you can make use of any ideas. There are so many simple ideas, but you can make use of the fish waste, or shellfish, or finfish waste. You can use for different purposes as a supplement to the protein, or use it as a live feed for the other organisms, or as a media for protein source for the feed, or you can use the calcium from that and show the kind of experiment in showing that the level of calcium. All these things can be done. So simple projects these are few projects I have tried to show you. Testing of plants with gravity or phototropism you can use. Testing of tea quality based on the fruit flies. You mix the fruit along with the tea. Then you can check whether the fruit flies are live or dead. Similarly, cleaning of oil from the birds, because most of the when the ship breaks or damage to the ship happens, oil tankers, they end up in oil spills, and these birds get affected. You can check what kind of oil spills, how we can degrade these oils, convert these oils. All these things can be done. And similarly, a simple experiment consisting of vitamin C tablets, you can give and see how this vitamin C tablet controls cold in one population, and one population is being given a placebo where it doesn't have any vitamin C, and how the population reacted. Coming to the evaluation, how we evaluate this project. Last year I was judging the competitions. These are the criteria on which the projects have been given marks. If you see, maximum marks are given to quantum of the experiment of work and creativity of the project and how the candidate present orally. These three takes up the 60% of the marks. We have to strengthen the kids here and try to design a research project which should have a clarity in topic. It should be a new work. It should be verifiable. We need to verify it, whether it is really done or just like that. Similarly, in case of scientific methodology, what kind of methodology is being used? Experimental plan, how the experiment is being executed, what are the instruments which are being used, where has been done. Similarly, variables, what are the variables, what is the control, whether the control has been defined properly or not. All these things matters. Similarly, coming to the quantum of the experimental work, it is how the data is collected. Is it reproducible or just and data cannot be reproducible? And is there any kind of analysis has been done on the data? Is there any kind of a statistics has been done on the data, or data support conclusions? How this conclusion is supported by the data? Data may say something and conclusion may be something else. These kind of things we have to keep in mind. And creativity or innovations, how creative is that project is. This is the crux of the science fair, where we try to kindle the creativity among the students so that they become innovators. In the written part, how this logical organization of the materials are there, figures have been properly handled well or not. Does it been supported by proper documents or proper references are being cited? All these things matters in the written part. In the case of oral, whether the students understand what he is doing. Most of the time what happens, it is been done by this teacher and the students present the work. So to avoid this, the student has a better basic understanding of that work, what he has done or she has done. Is he or she is able to interpret the results, what kind of interpretations they give, and have they done it independently or with the help of anybody else. The student may be able to maybe presenting her work, and from the sides his his friends or his teachers maybe giving clues when the candidate presents the results, during oral presentation. Is the student understand the questions, is able to independently handle the questions raised by the judges also matters. And does he had any kind of idea what kind of future work can be carried out in the field or the project? All these things matter when it comes to oral presentation. So with this, I stop here. If you have any doubts or any kind of things, please don't hesitate to ask. Sorry, my voice was very feeble to you. I have to address this issue, inshallah, in the next talk.
Thank you, sir. Now it is open to all. You can ask any question. It's a very good opportunity. You can clarify your doubt from the expert. You can unmute and talk. Any question?
Professor Nasir Bhai,
Yes, yes.
Qazi Azhar sir is here. He would like to talk, I think.
Yes, yes.
Maybe a few words.
Yes, yes. I requested Dr. Qazi Azhar sir to say a few words.
Assalamu alaikum.
Walaikum salam, sir.
Mashallah, it was a very good presentation, and from a research-oriented professor, I'm very happy. I'm sure there must be some questions addressed to him, because always teachers and the students ask about the ideas of the projects, and he has given quite a good examples. Of course, they have to focus on one of the projects. The other thing he has mentioned that we only accept projects which follows scientific method or engineering method. So that is most important. Otherwise, the project will not be accepted. And actually, when he was talking, I was smiling. He said that sometimes teachers do the project and the students memorize the things, memorize the project, how it was conducted and come to presentation. At that time, when you ask them questions, they are lost. And sometimes I have seen this very often in India, that projects are in English. Of course, the posters are in English, and the person who has done, or the student who has done the project may not be fluent in English. Yeah. So what they do is they give that project to some other student who can memorize better and speak good English or better English. That also creates problem.
Yes, we are not looking for language, but the
Right.
scientific interest.
It is not.
Yeah, we are looking for content of the project. So that pulls down that project, and the project may not be given, any award. So we have to be very thoughtful about these things, particularly teachers. And teachers can give idea of a project, but the student has to do it. Parents can give idea to the students, but the students have to do the project. Professors can give, they can go to the college and, one of the professor can give idea, but the must thing is student have to do the project and they have to know in and out of that project. And see, that's the thing. If the student does the project, the student will know what are the things went wrong and how he corrected in the project, and how the data was not reflected initially, and when he kept on repeating the project, and the data was very supportive, or may not be in favor of the hypothesis, and the students can debate on that, can argue with that, and then the judges will come to know that yes, this is the person who actually did the data, collected the data and interpreted the data, analyzed the data. So those are the things which are very important. And once again, I thank the presenter. He's very, voice was a little bit volume. I increased my volume to listen to him. So no, it's okay. You did a wonderful job and congratulations. And I will leave rest of the things to ask if the audience have any question. Thank you.
Any question from the teachers? Feel free to ask. Okay. You can unmute and ask, or you can put in the chat. Always there is an inhibition to raise question.
It means only two things. They have clearly understood and not understood.
Normally, as good as students know, even teacher is seated to raise question. Alhamdulillah, Dr. Shahid Naveed, Alhamdulillah, gave a very good talk, and he covered everything: how to select a topic, how to do a project, how to present a project as a research paper, how to orally present, and he also covered what is the expectation of the judges. To that extent, he is clearly present. As far as the main part of the project is concerned, that is the experimental part. What Sir insisted, I would like to re-emphasize. One need to understand the controls in the project. That is, need to understand the dependent variable, independent variable. Particularly doing the biological project and botany project, one has to have the reference control, and that is very important. Sampling is very important. You need to repeat whether the procedure works. You need to repeat the project as well. Sir listed a various topics in various areas: microbiology, botany, environmental. Various topic is suggested, and what projects can be done in each topic, as well as he gave some sample projects also. I hope the session has been very useful. Inshallah, everyone will come out with a very good project this time, and let the project be innovative. Let the project be your own original project, and inshallah, and let the student do. You can give the idea as a teacher, you guide them, and inshallah, let us expect a very good project to come up for the coming National Science Fair in January 2023. And we will have the one more session in the series. Next week, Dr. Shah will speak in Urdu, and myself will talk in Tamil. So regional language. We both will present in next week. The next week the session will start by 4:15. Uh, it will go till 6:15. I hope you all join, and particularly we expect the schools from the rural villages are to join, as well as we expect the schools from the North Arcot, Ambur, where the uh, Urdu is mother tongue and spoken. Those schools in large number we expect to join. We wish to reach the even interior of the Tamil Nadu. That is the reason we are conducting. First time we are going to conduct in regional language. You spread the news. You too join, and you tell your colleagues and other schools. Let many in large number join in the next week. Now, over to Dr. Nihar Akhtar, our Joint Secretary, OMIAN, to propose vote of thanks.
Assalamu alaikum wa rahmatullahi wa barakatuhu. I wish to thank Brother Saquib Naveed for giving an elaborate lecture on how to present the research papers and going into very a detailed manner. Since he himself is a researcher and a reviewer, we had the benefit of listening to him and fine-tuning our work. Mashallah, it was a very wonderful session. It was very informative. Thank you very much, Dr. Saquib Naveed, brother. And I wish to thank Dr. Hazi Azhar, all the way from USA. In the early hours, he is sitting with us, and he has been our mentor. And either he is physically present or virtually present, or his duas are always there in all our science fairs. Thank you very much, sir. Jazakallah khair. And thank you, Dr. Abdul Kamal Nazar, for organizing the series of lectures on science fair and taking the science fair very seriously and putting in a lot of effort in the science fairs and inculcating scientific temper in children. May Allah reward you abundantly, sir. And finally, I would like to thank all the teachers who are present here, and also my colleagues at Omiyat, Brother Fazal and Brother Syed Salim. Thank you very much, sir. And as sir was saying, next session will be at 4:15. Please come in large numbers. Yeah, and many of you are primary teachers. We hope to have plenty of primary school projects this year, inshallah. Thank you very much.
Thank you, madam. Thank you.
Jazakallah khair,
Thanks for the
Nihar
opportunity.
ma'am. Thank you.
Sir, no?
Alhamdulillah. With this, we are ending the session. Jazakallah khair. Assalamu alaikum. Good evening to one and all.
Thank you, sir.
Wa alaikum assalam.
Other person?